Hi guys, today we're going to be talking about the chemistry behind chemotherapy, and we'll go through 3 different examples of specific drug types.
38% of people will be diagnosed with cancer in their lifetime. The leading treatment for cancer is chemotherapy.
The main goal of chemotherapy is to kill the cells in the body of someone diagnosed with cancer so that their body can regenerate new, non-cancerous cells. There are many methods that can be used to reach this goal. Each cancer patient is different and requires many types of treatments. The 3 main types of treatments that we are talking about in this presentation are alkylating agents, mitotic inhibitors, and topoisomerase inhibitors. The first type of treatment that I will talk about is the use of alkylating agents. Alkylating agents are some of the first chemotherapeutic drugs created and are still used to this day. These drugs work at the molecular level by binding to negatively charged sites on DNA such as oxygen, nitrogen, phosphorus, and sulfur atoms. An alkyl group is any group with the formula CnH2n+1. So for example, a methyl group, propyl group, or butyl group would all be considered alkyl groups. This process is called alkylation because the result of the chemical reaction is a DNA molecule with an attached chain of carbon atoms. The action of alkylating agents as chemotherapeutic drugs comes from their ability to irreversibly bind to DNA, and once bound, the altered molecule disrupts the normal action and replication of the DNA strand. The 7th nitrogen atom of guanine is especially vulnerable. The agents promote intra and inter-DNA strand linking at guanine bases on the strand. If you look at the image, you can see the intra-linkage or on the same strand, or interlinkage or on the opposite strands. This leads to either programmed cell death, apoptosis, or at least an arrest in cellular replication. One of the most used classical alkylating agents in chemotherapy is called mechlorethamine. It is used primarily for lymphomas, which are cancers of the lymph nodes. The N-chlorine atoms bind to the 7th nitrogen on 2 different guanine bases and permanently link them together. If you look at the image on the right, you can see the chemical structure of the drug. If you look at the image on the left, you can see how it works once introduced into the body. Once the drug enters the cell, it binds to the 7th nitrogen on the guanine molecule from each strand, and on the right of the long blue arrow, you can see what the final linkage of the 2 guanines would look like in the cell. One of the most used non-classical alkylating agents in cancer treatment is cisplatin. It's called a non-classical alkylating agent because it does not have any alkyl groups in its chemical composition, but functions in the same way as classical agents. If you look at the image, you can see that it consists of 2 NH3 groups, a platinum, and 2 chlorine atoms. Similarly to the classical agents, these drugs still bind 2 guanine bases together at the 7th nitrogen with 2 chlorine atoms and permanently prevents DNA replication in that cell. The drug still binds the 2 strands either together or in opposite strands. Also, if you look at the image of the guanine atom at the bottom, you can see why the 7th nitrogen is so susceptible to binding with the drug. It's so willing to bind with the chlorine atoms because it has a lone pair on it. Alkylating agents have limitations. Their functionality has been found to be limited when in the presence of the DNA repair enzyme 0,6-methylguanine DNA methyltransferase, or MGMT. Cross-linking of the double-stranded DNA by alkylating agents is inhibited by this DNA repair mechanism. If the MGMT promoter region is methylated and therefore no longer transcribed in the cells no longer produce MGMT and are therefore more responsive to the alkylating agents. Methylation of the MGMT promoter is a useful predictor of the responsiveness of tumors to alkylating agents. If you look at the image, you can see the chemical structure of MGMT.
So the next class of chemotherapy drugs that we will be talking about are mitotic inhibitors. Mitotic inhibitors work by slowing or stopping the growth and spread of cancer cells. They are used to treat a wide range of cancers, including breast cancer, ovarian cancer, and testicular cancer. To learn about the mechanism behind mitotic inhibitors, we will focus on one specific type called vinblastine. Vinblastine comes from the vinca plant pictured and is poisonous to most organisms. Vinblastine works by binding to the tubulin used for microtubules and inhibiting stabilization. By doing so, it prevents mitotic spindles from pulling chromosomes into their new cells and dividing. This arrests the cell in the G2M phase of the cell cycle and causes cell death or cell apoptosis. Vinblastine is used mainly in lung cancer and lymphoma. Before we can understand how vinblastine works on the chemical level, we have to learn how the microtubule is chemically structured. The microtubule is made up of biopolymers that form through the polymerization of heterodimers of the negative alpha and positive beta tubulins. The negative end of the alpha tubulin will bind to the positive end of the beta tubulin over and over, creating the stricture pictured below. This individual polarity will create an overall polarity for the microtubule, which is essential in its process of disassembly and assembly. The process of disassembly uses the GDP molecule to break the biopolymers apart. The process of assembly uses the GT molecule to reassemble the individual heterodimers. Both the process of assembly and disassembly, also known as catastrophe and rescue, are essential to a properly functioning microtubule. The microtubule is essential in the function of mitotic spindles and thus cell division. So now that we understand how microtubules work, we can understand how vinblastine prevents them from functioning. Vinblastine binds to the positively charged beta tubulin, and in doing so, prevents the GTP molecule from binding to that site. The GTP molecule is used to assemble the microtubule, as we already know, and by blocking its binding site, vinblastine prevents the assembly from occurring. So, if the microtubule cannot polymerize, then the mitotic spindle will not work. As you can see in the cell cycle pictured, it is the job of the mitotic spindle to pull the chromosomes apart. If vinblastine inhibits this function, then the cell cycle will stop and the cell will go into cell apoptosis, which is also known as cell death. If cells are unable to duplicate, then cell growth will slow down and eventually stop, killing off the cancerous cells in an individual's body so that they may eventually regenerate new, healthy ones.
Another form of chemotherapy is called topoisomerase inhibitors. These drugs are chemical compounds that prevent the action of the enzyme topoisomerase. During the cell cycle, topoisomerase is an enzyme used in DNA replication specifically. It catalyzes the winding and unwinding of the phosphodiester backbone of the DNA double helix so that it can be replicated more easily. Cells need this enzyme to keep their DNA in the proper shape while they're dividing. Therefore, if topoisomerase is damaged during the cell cycle, the DNA cannot be replicated and the cells cannot duplicate. Also, there is topoisomerase 1 and 2. On the top right, you could see the topoisomerase 1 structure, and on the bottom, you could see the topoisomerase 2 structure. Topoisomerase inhibitors are the drugs that break down the enzyme just enough so that it stops its function and eventually kills the cell. They are also known as topopoisons. More specifically, they interfere with the religation step of the catalytic reaction in DNA replication. This results in DNA damage accumulation and leads to the breakage of the single and double strands in the DNA helix. Then, unrepaired DNA damage signals cell cycle arrest, which signals programmed cell death, also known as apoptosis. If you look at the image on the right, you can see a comparison of how topoisomerase 1 and 2 interfere as the enzyme travels down the helix. Eventually, both inhibitors lead to cell death. Looking more in depth, under normal conditions, topoisomerase 1 and 2 both cleave and religate DNA very rapidly, as you can see in the image from A to B and from E to F. Topoisomerase inhibitors in the chemotherapy, as seen as green in the animation, bind reversibly to the interface of the cleaved DNA and the enzyme by forming a ternary complex. This works because the inhibitors simultaneously interact with the DNA and the enzyme. The topoisomerase enzyme forms specific amino acid contact with the drug because it thinks that it is stacking against the bases. The drug can then interfere with the bases and cause mutations that force the cell into apoptosis. Irinotecan is a specific type of topoisomerase 1 inhibitor. It is used in the treatment of colorectal, small cell lung, ovarian, and hematological cancers. It works in the body like most other chemotherapies. In the image, you could see the chemical structure of irinotecan. Although all chemotherapy has its disadvantages, topoisomerase I has been a useful link in the treating of cancer because the enzyme is usually found in rapidly duplicating cells, such as cancer cells. This can link the drug specifically to cancer cells instead of attacking all the cells in the body in the same way. The process that irinotecan goes through in order to treat cancer takes about 5 steps. First, topoisomerase I binds to DNA. Second, the enzyme begins to de-stress the strand and creates single-stranded breaks. Then, irinotecan binds to topoisomerase I, as seen in the image, and prevents religation of the single-stranded breaks. Then, the topoisomerase inhibitor and the enzyme approach the replication fork in the DNA and interfere with the breaks in the DNA. Finally, the cell signals for apoptosis due to its mutations. In the image on the left, you can visualize how irinotecan attaches to the topoisomerase enzyme and eventually leads to apoptosis. In conclusion, all of these chemotherapy drugs have the same goal: to prevent cancer cells from dividing and multiplying. 3 types of chemotherapy that we talked about were alkylating agents, mitotic inhibitors, and topoisomerase inhibitors. Aside from these 3 chemotherapy drugs, there are a multitude of others. In a world with increasingly more personalized medications, there have been a shift to treating cancer with gene-specific drugs known as gene therapy. One point you may have noticed is that all these drugs target cells in general, not just cancer cells. This means that when these therapies are used, all the cells in the body are targeted, which is why chemotherapy is such a debilitating treatment option. However, by killing cells in the body, the cancer will also die off. When they die off, it gives the body the opportunity to produce new, non-cancerous cells. The advantages of treating cancer with chemotherapy are that there are many different types of chemotherapies to choose from. However, a lot of these drugs can actually pose a risk to getting cancer for a second time because they contain carcinogens. Thank you for listening. This has been Jen, Julia, and Eric from the Honors General Chemistry 163.